Thank you very much for the opportunity to present at this meeting. My name is Holger Fröhlich, and I am from the Fraunhofer Institute for Algorithms and Scientific Computing. Let me start with an overview about the current situation in the biopharmaceutical industry. As you can see from the figure on this slide, the efficacy of pharma R&D is declining continuously over the last decades. Nowadays, more than 90% of clinical studies fail, with corresponding huge economic losses. Moreover, in many countries of the world, there's additional pressure because refunding of approved drugs by the healthcare system requires to demonstrate an added value compared to pre-existing therapeutics. In consequence, the biopharmaceutical industry is seeking a lot for innovative solutions to improve the situation. What do we perceive as a need for pharma industry to improve the situation? On one hand, we see the need for intelligent approaches that yield faster and more efficient development of drug candidates, that means candidate molecules. On the other hand, we think that there's a high potential for approaches that aim at enhancing the success chances of clinical trials. Notably, the failure of a clinical trial could be due to different reasons. One, choice of the wrong target disease mechanism, which results into low efficacy or unfavorable side effects. Two, selection of the wrong target population. To illustrate the last point, let us have a look onto the right-hand side of the slide. Here you can see a figure taken from a Nature publication by Holger and co-authors from the year 2015. It shows the fraction of patients benefiting from a given drug in blue versus those not showing a clear benefit in red. This figure demonstrates that not every patient responds to a given drug at the same extent. Hence, we need approaches to preferably select patients for a trial that are likely to benefit. That means we need to bring the right drug to the right patients. This is exactly the vision of precision medicine. To realize solutions for the points mentioned before, we need informative, large-scale, and high-quality data. These data need to cover the entire biopharmaceutical value chain, and it has to be collected systematically. To analyze the data and to guide decision-making, we need modern techniques from data science and artificial intelligence. These techniques can be applied at every step of the value chain, as demonstrated on this slide. A darker color indicates a stronger perceived impact on business. Within my team at Fraunhofer, we aim for realizing the potential of AI and data science in the fields of precision medicine on one hand and early drug discovery, specifically systems medicine, on the other. We develop our solutions jointly together with partners in pharma and biotech industry as well as hospitals. I like to point out that questions in medicine and according requests by our partners are very complex and heterogeneous. Generic off-the-shelf solutions are unlikely to be successful. Hence, we develop and deliver specifically tailored methods and solutions covering a variety of techniques, such as hybrid AI, time series modeling, and multimodal, multi-scale data fusion. Our newly developed methods are always application-oriented. Our work thus forms a bridge between academic and industrial research. Altogether, the work in our team is well aligned along the value chain in biopharmaceutical research. It covers AI approaches for biological system reconstruction and modeling, drug targets identification, bringing the right drug to the right patients, which is the core of precision medicine, and finally, also simulation methods for clinical trial design. Let me now provide you with a few more detailed examples of our work. We have developed dedicated AI approaches which allow us to combine different types of experimental data, such as gene expression, with a knowledge graph representation of disease biology. Our methods allow us to quantitatively simulate interventions into the biological system, for example, via a drug. Hence, we can estimate the effect of a therapeutic intervention on the disease phenotype, as well as on relevant biological mechanisms. Moreover, we can prioritize drug targets. Altogether, our AI approaches could thus yield better informed decisions on target selections and thus help to make clinical trials more successful. Noteworthy, our approach has already been tested successfully by our partners in several indication areas. Let me now explain you our AI strategy for bringing the right drug to the right patients. Specifically in neurodegenerative diseases such as Alzheimer's, it is critical to diagnose the disease as early as possible in order to have a chance with any sort of medication. Hence, the first step of our strategy is to develop AI-based personalized risk models that allow for an early detection of individuals at high risk for a later disease diagnosis. Importantly, these models consider the characteristics of the individual patient, rather than only relying on population averages. The second step in our strategy is the AI-based stratification of diagnosed patients into sufficiently homogeneous disease subgroups. For that purpose, we consider molecular mechanisms as well as multiple clinical outcomes. Stratification of patients into subgroups can be utilized in the context of clinical trial design. For example, by considering the grouping of patients in the statistical analysis of clinical outcomes at the end of the study, or via an adaptive clinical trial design. In such a setting, information about strata, together with observed treatment responses, could be used to narrow down the trial to the most responsive subgroups. The third step in our strategy is the AI-based simulation of disease trajectories of individual patients under different inclusion and exclusion criteria in order to optimize clinical trial design. Importantly, the first three steps can be performed with the help of publicly available cohort studies, which have been conducted in many areas of disease. The last step is to develop AI models for predicting clinical treatment response of individual patients. It aims for identifying likely responses of a drug early during a study, ideally already at baseline. This last step requires sufficiently large data from interventional clinical studies, of course. A possible strategy is to enlarge phase 2 trials and use these data to build models. Subsequent phase 3 trials could then be much smaller and only focus on those patients that are predicted to be likely responders according to the AI model. In summary, all 4 steps of our strategy aim for optimizing clinical trial designs via different approaches. To illustrate our strategy on the last slide, let me first show you an example of some work that we did in the field of Alzheimer's disease. In this work, funded via the private-public partnership project Autonomy, we developed a personalized AI-based risk model. The model has been trained on data of cognitively normal and mild cognitively impaired patients from a well-known cohort study in Alzheimer's disease. And it has been externally validated on another study. It is the first retrospectively validated AI-based risk model in the Alzheimer's disease field according to our knowledge. The AI model predicts the time-dependent risk of an individual person to receive the clinical diagnosis dementia. Each of the curves shown in the plot in the middle depicts such individual risks. Altogether, the model allows for an accurate detection of high-risk patients about 6 years prior to clinical diagnosis. It could thus be used to recruit patients into clinical studies already in the mild cognitively impaired stage. The second step of our strategy comprises the stratification of patients into homogeneous subgroups. As one pillar in this strategy, we focus on molecular mechanisms. This is because it is now commonly believed that the purely phenotype-based definition of diseases stemming from the 19th century is likely outdated and should be replaced by a definition of diseases and disease subtypes based on molecular mechanisms. An according call for a reform of disease taxonomies has already been published a decade ago by pharma industry leaders. The EU-wide private-public partnership project Etonomy was largely a response to this call. Within Etonomy, a project coordinated by our institute, we demonstrated for the first time that it is possible to integrate knowledge about molecular disease mechanisms with genomic data into AI algorithms that allowed us to reproducibly identify molecular disease subtypes of Alzheimer's and Parkinson's disease. We could show that these disease subtypes manifest into distinct clinical, pathophysiological, and molecular differences between patient groups. In addition to systematic molecular differences between patients, there's also a highly varying progression of disease, as indicated on the left-hand side of this slide. This imposes a high challenge for the success of clinical trials in many areas of disease, such as Alzheimer's and Parkinson's disease. Noteworthy, the progression of many diseases is only partially driven by genetic factors, because environment, lifestyle, and comorbidities may also have an influence. This suggests to identify progression subgroups that exhibit a similar pattern of disease progression. In many areas of disease, there's not just one way to measure disease progression. For example, in Alzheimer's disease, a variety of scores have been developed to measure different aspects of cognitive impairment. Accordingly, we have developed a novel AI approach that allows for identifying disease progression subtypes by looking simultaneously at different clinical outcomes. In addition, we have developed AI-based classifiers that allow for predicting the most likely progression subtype for the individual patient already at baseline. Accordingly, our stratification can be used within an adaptive clinical trial design, or by considering the patient subgrouping in the statistical analysis at the end of the study. Finally, there's the possibility to preferentially recruit patients of a specific subgroup at start of the study. The third step in our AI strategy for bringing the right drug to the right patients is the simulation of patient trajectories. Once again, this work has started as part of the private-public partnership project Etonomy, and in the meantime, it is also part of the German National Infrastructure for Health Data, NFDI for Health, abbreviated. The idea is to learn a generative AI model based on data from a real-world longitudinal cohort study. For this purpose, we have developed dedicated AI methods. These methods result into an abstract mathematical representation of the real-world data. That means they learn a kind of rule behind the data. Our methods that allow to simulate virtual subject trajectories up to an unlimited quantity. Of course, the immediate question is how good these simulations are. Hence, we perform a rigorous quality assessment which measures the goodness of fit between synthetic subjects and the regional real-world cohort. What is now the value of having sufficiently realistic simulations of synthetic patient trajectories? Well, they can be used for optimizing clinical trials. More specifically, we can simulate what-if scenarios, for example, a counterfactual modification of baseline characteristics of patients, such as disease severity and age. Accordingly, our model can simulate how clinical endpoints at the end of the study might change. Hence, we can use our model for optimizing inclusion and exclusion criteria, or for estimating the statistical power for detecting differences between defined patient groups. Finally, our approaches also allow for extrapolating beyond the end of the study. Let me illustrate the concept on the previous slide with an example. In our work, we have developed an AI technology, which we call WAMBEN. This method allows for modeling entire clinical studies in a longitudinal manner. WAMBEN is a white box model. It can be visualized as a network, as shown in the top right. The network depicts dependencies between different variables and variable groups in the data and assigns each of these dependencies with a certain level of statistical confidence indicated by the little numbers on edges. WAMBEN allows for simulating what-if scenarios via well-established theory. It thus allows for simulating consequences of modifying inclusion-exclusion criteria. For example, the two right-hand side plots on the bottom demonstrate the simulated effect of altering disease severity at the study's screening phase on a major clinical outcome later during the study. Likewise, the right plot shows a similar simulation for a counterfactual change of age at study baseline. The final step of our strategy is the development of AI models for predicting the clinical response to a given drug. For this purpose, we have developed an AI approach that integrates genomic and clinical data of an individual patient. Regarding genomic data, we focus on the disease etiology as well as drug mechanism of action. Based on this knowledge-driven approach, we can handle even whole genome sequencing data for only a few hundred patients. Using our approach, we have recently developed the first retrospectively validated AI model for predicting the clinical response to an anti-epileptic drug. Our model allowed us to better understand the non-response to the drug, as well as estimating the anticipated effect on sample size for a clinical trial that would preferentially recruit likely responders. Our approach can be seen as a blueprint for similar projects in the area of precision medicine. It could be adopted in a strategy that enlarges phase 2 trials to build models and subsequently focuses the recruitment in phase 3 on likely responders. This would dramatically reduce sample size in phase 3. Let me summarize. AI and data science are now widely seen as essential to support biopharmaceutical R&D. At Fraunhofer, we have a long-standing experience with the analysis of health-related data of various kinds. Based on these data, we develop problem-specific algorithms and solutions along a value chain that ranges from biological system reconstruction and modeling over drug target identification, finding the right drug for the right patients, up to simulation of synthetic cohorts for optimizing clinical trial design. We work closely together with many national and international partners, either directly or within third-party funded projects. If you're interested to work with us, I'm happy to discuss further with you. You can find my email at the bottom of this slide. Thank you very much for your attention.